I'm really interested in the visual system, how the pattern of light that falls on the back of our eye is changed into something that we perceive and sometimes even think about. So that's the big question that drives my research. I'm really interested in thinking, so I want to know how thinking works. And some visual tasks really require you to look at it and really think what you see. And that's the thing that I find most fascinating to work on that. We typically use tasks in which humans, monkeys, mice have to look at the stimulus and they have to work it out. For instance, one task that we have been using a lot is one in which you have a couple of curves that run through the visual field. And the animal or the human has to see what is connected to what. And that takes a lot of time, allowing us to study what happens in the brain when you try to figure it out. In the beginning, I thought this will never have any use in society. I changed because now we're working on a project in which we try to implant many electrodes, so up to 1,000 electrodes now in a monkey, in the visual cortex. If you stimulate one electrode, the animal perceives a dot of light. It's called a phosphene. Since we have 1,000 electrodes, you can see 1,000 dots of light at different locations in the visual field. And we use the system as a sort of matrix board that you know from along the highway. If you light up one bulb on the matrix board, you see a dot. But by lighting up a pattern, you can see a shape. And we think we can use this for blind individuals to later help them give a visual cortical prosthesis. So for those individuals whose eyes are no longer working. But that's a very ambitious thing, and it will take some time before we really bring it into a clinical product. I thought it was terribly exciting. So I just want to know how consciousness works and how perception works. I'm actually trained as a medical doctor, so the medical questions were always in the back of my mind, but I thought I was just going to study basic research, basic questions, like what does it mean for a stimulus to reach consciousness? Yes, as a medical student, I already started to study neural networks, and that came in handy later. Now, as we're working towards making a visual cortical prosthesis for the blind, I have to dive into unexpected territory. So I'm now sometimes looking into machine learning. I'm also very interested in materials, so we want to build a prosthesis that is safe and that will be durable. So if you implant it now, it's still working, say, in 2 or 5 or 10 years, and that requires knowledge also about materials, and that's what I have to study now. So in my own research, I have a couple of heroes. Nikos Logothetis is one of my heroes. And one study that influenced me a lot, and I think many of my colleagues, is when he demonstrated that neurons at different brain regions report whether a stimulus is in the consciousness of an animal, say a monkey, or not. And that was really, really important, I think, for everybody in the field. And then he has also done tremendous work on understanding the basis of functional imaging. I think the main big question that also drives me, but it will keep driving generations, is you have, for any task, you have activity across so many brain structures. Some of them are in the cortex, some of them are in the subcortex. So how do all these systems work together? Because if something happens, say you become conscious of something, this is not something that is restricted to cortex. You see it across many, many structures. How do the brain cells and all these structures work together and try to distill a coherent percept, a coherent thought, out of this, at first sight, somewhat random activity of these neuronal spikes? Difficult question, because we are at the relative start. But so far, I think that people are converging on the idea that you have to do rigorous studies, and you need the effort of many, many people in neuroscience using many different methods to really understand the brain in all its facets. It's a big honor to be here because for me a lot of heroes are here. So the people who drove systems neuroscience in the last 30 years or so are here, right? And these are the people who made the breakthroughs possible that now allow us to understand a little bit of what it means to attend to something or when something enters into consciousness, these kind of big questions. The people here, they are ones that gave big contributions to answering these questions. I like both. So I like this type of meeting because you really get a chance to talk to your colleagues and pick up on new developments, or hear about what's going on in their labs, exchanging knowledge about different techniques. At the Society for Neuroscience meeting, you really have to do very careful planning because it's huge and overwhelming, and your contact with your colleagues is much more superficial. On the other hand, it's very rich, so you can get a lot of information in just a couple of days.